Welcome, and my name is Yunlong Liu. I'm a faculty member in the Department of Medical and Molecular Genetics. Let me see whether this works. I hope so. All right. And I'm also a member for the Center for Computational Biology and Bioinformatics, a member from Center for Medical Genomics. And the CCBB, Center for Computational Biology and Bioinformatics, located in the 5th floor here. So that is also my office hours and place. And the Center for Medical Genomics is the core facility for the IUPUI campus in terms of next generation sequencing technology. So we will have more information regarding that. Okay, I think so many of you are here, and that really speaks for itself. This is a very important technology, next generation sequencing. So if you go to any national conference right now, regardless of which disease, which biological system you really study, and it's hard to skip this term. I think it's a good choice that you want to learn a little bit more about this, and for both of your research and your future career development. And my lab has started to know this technology from 2007, and at that point that I haven't heard about that until one collaborator comes into my office, can you do this analysis? And my response is, of course we can, although I haven't heard about it. And back in those days, we really are pretty naive about things. We start to do the blast and all those kind of things. As the course progress, you will see that is that's not going to make it. And during these past 4 years, we really learned a lot, and both from the technology-wise and from the informatics-wise, and most importantly, that we made so many mistakes that I want to introduce in the class, so you don't have to make those again. So I think it becomes, this is my first time to have this class and to have this course, and all the information, the feedbacks are welcome, and I will try my best to keep it up, and I don't know how well or bad I can do, but so bear with me. And as I'm trying to prepare this class, I think I'm the one that get the most benefit, and I learned so much systematically, and reading a lot of papers regarding this technology. I really figured out a lot of things that I would never figure out. So I think it's a good thing for myself as well. All right. Speaking of this technology, next generation sequencing technology, just give you a little bit heads up if you're not familiar with this. So we know that we had a human genome project. That is in 1990 to 2001. I think the first human genome project, the publication came out in 2001. And back in those days, that you can calculate it, they spent 12 years, and hundreds of labs in the world were working on that. And they spent $3 billion and then figure out one draft of human genome. And that was amazing. That's the first time that we can do that. But today, that with the same amount of time, I don't know how much we can get done, but if we only focus on 1 week of work, on one equipment, one technician, we can get 2 drafts of human genome done in a week. So that is how technology development really can promote this science. And so far for today, the cost for one genome is about $10,000 rather than $3 billion that hundreds of labs struggle with. Okay? And before we get into it, some of the logistics about this class, and the time will be now 3 to 6 p.m. And I just trying to select the most inconvenient time for you. So make you struggle and do not go to bars. Although the location is nice, so we have the bar just outside. If after the class that we want to go there, and that's a good place to go. And I will be the major instructor, so most of the class will be on me. And we do have very exciting guest lectures. And Dr. Howard Edinburgh, a professor from biochemistry department, and he and Dr. Xiaoling Xie are from the Center for Medical Genomics, and they will have a guest lecture to introduce the library preparations for the solid platform, and more excitingly, you will have the opportunity to tour the core facility to look at those big machines. And that is nice. And Tatiana Freud will talk about the exon sequencing and using the disease sample as example. And Dr. Bill Muro from Purdue University will also, and he is a population geneticist, statistical geneticist, and he studies chicken and how to change the chicken genome to make them grow more meat, I guess. And he will come here to give a very interesting guest lecture. And also Milan Rakovic, and who was a graduate student not long ago, graduated from our department, medical genetics, and now he's a faculty member in Department of Surgery. So the office hour is Fridays morning. So I was just trying to block out all my Fridays. And from 9 to 11 a.m., the location will be in my office up 5th floor of this building. And here's my email address and phone numbers, okay? And there will be no textbook, okay? There will be no textbook. And I tried to find, and there's nothing relevant. So we'll try to get a lot of papers accumulated. So you will have the opportunity to go to OnCourse system. There's a lot of papers will be uploaded there, and you feel free to read those, okay? And this is the most exciting slides, all right? So we got a TA, Hao Yu, and he, can you show your hands? Yeah. So, and he is a School of Informatics student, and so he will be answering all your emails, all your technical questions, and grading your homeworks and final exams. So if you didn't get good enough in the final exam, and he will be the one to be killed. Okay. Yeah, don't worry about that. He's not going to do any of those. So I will still be responsible for answering emails and grading those homeworks and assignments. So I'm still the one to be blamed. And the Hao Yu's role in this class is he will help us to do more coordination. And we already have 24 students registered, so that's already a pretty big class. And not to say there's so many people who are interested in sitting in. And so he will upload the materials in the OnCourse system, most importantly, finding out all those materials, the papers that we are going to cover in each lecture and put there into as a resource part. And there's one assignment already for Hao Yu, because there's so many of you are interested in sitting in. So if you can send him an email and you are interested in attending this, and he will add you into the OnCourse system as an observer, so that you have the access to all the slides and other information and the papers there as well. And I'm not going to give you grades or anything like that, but you will have full access to all the resources that we'll be distributing. If you are not an IU student or employee, you don't have an IU account, and also send an email to him, and we'll figure out a way to set up a guest account so that you can still, to a certain extent, get access. But those are the things I haven't figured out. We need to do some testing. So I expect he will receive a lot of emails in the next couple of days. And the course objective is to understanding the basic principles of next-generation sequencing technology. This includes... So you can hear me well from the back, right? All right. This really includes the basic biological applications. And so by the end of the class, I expect you to know what this technology is going to help us, where it's going to help us. And there's many different applications. There's RNA sequencing, DNA sequencing, and it's my hope that you will be getting familiar with those. And I want you to understand the basics of the data processing, okay? And meaning that you will be able to move data along. So once you get the data, the raw data, terabytes of data from the core facility, and you don't even know which software you should use to open it at that point, I expect you can start from that point and going forward and having basic information and basic softwares that you can use and trying to go through the data analysis. And for the statistical and informatics theory part in the data analysis, and for example, it sounds simple, but it's actually not. You got the DNA sequence, and how can you figure out there's a variation there? Okay, and there's a lot of statistical theories behind that. I'm not expecting you understand all of those because I don't, and I'm not expecting you to develop novel things. However, I want you to understand the advantage of and the limitations of each individual methodology, and also, most importantly, the assumptions of different methodologies. So I can give you one example. We know that chip-seq experiment, which I will have a couple of lectures to cover that. And if you don't know the assumptions of different algorithms, and it's very possible that you did some experiment, you just try some Googling and you see, oh, this is the software, it looks pretty good. And you use that, and it turns out to be the peak identifier, which is nice, and to study transcription factors. However, your data is a histone-modification data. So RNA polymerase II, it doesn't supposed to form those peaks. And if you don't know those assumptions, and you probably will not be able to use that very effectively. So I'm expecting that this is very important. You want to know the assumptions of different algorithms and different methodologies. And the biological interpretation of the result is very, very important. So of course, you will have to really know about it. For those of you who registered the class in the course evaluation, okay, and we will have in-class quiz, okay, depends on my mood in the morning, so it will be taken randomly, and that will be the 10% of the final grade. And the reason, and it usually will take taken at the beginning of the class. Oh, by the way, the cell phones, please turn those off, and otherwise you are responsible to bring in donuts next time. That's another penalty. Right, and that is just trying to make sure the registered student, at least you come here on time. All right, and we will have homeworks, and again, I haven't figured out which one to do, but the requirement for this is really computer-based practice. So if I talk about alignment next time, I will expect you to be able to use certain software to do it, and I can give you some sample data, and you should be able to do it. And those are part of the homeworks, and it will be 20% of the final grade. Project, okay? We have 24 students registered class, 12 from School of Informatics, 12 from School of Medicine. And I want you to form 8 groups. So 3 students each group. You just, these are only for the registered students, I'm sorry. And you want to randomly find your peer in your group, okay? And of course, you can find the ones that you like. And alternatively, probably it's more wisely, you find the ones that which has a complementary background with you. So if you're a biology student, you want to find computer science students in your group so that you can make some project and from different perspective. And the project itself will be 30% of your final grade. And there's a couple of deadlines I'm designing here, and September 23rd will be the day that you want to fix in your topic. You want to discuss with me before that, and whether that topic is good. And I want you to update the progress every 2 weeks, and we will set up time on that. And the final report is due on November 28th. I think that's just after Thanksgiving. If you want to do it before Thanksgiving, let me know. I can change the deadline. And the final presentation will be December 2nd. And the 40% will be the final exam, and that will be on December 9th. Okay? Any questions so far? These are the logistics. Yeah? Is the exam open book or closed? Oh, it's open everything. So not open to your neighbors, and other than that, and it will be open book. Yeah, you can open to internet if you want. Yeah. All right. Any other questions? No? All right. So next one is I want to give you a brief introduction of the course overview, and I think this is particularly important for the people who are sitting in, and you probably don't want to do all the lectures, and depends on your interest. So I assume that most of you know where the course website is. You just go to my webpage, and there's a link there, so you can go to the course website. And today is lecture 1, so we will talk about the next generation sequencing. It's an overview. And the second part, next lecture, will be more on the data processing part. So for the people, for the biologists who are sitting in, not for the register class students. So if you are not really interested in very single detail of this analysis, I think that is a lecture you can skip. And I'm going to talk about a basic analysis workflow, the sequencing quality evaluation, alignment theories, and I'll spend a lot of time on that, and you may or may not be interested in. And the data formats goes very detailed, what data is available in different file formats, and data visualization and things like that. And after that, we will have 3 lectures to talk about the DNA sequencing applications, including most biological applications. Okay. I'm going to accept, that's fine. Biological applications, and we will have 2 lectures regarding the bioinformatics topics related to that. So September 16th are the day that we will have the tour for the Center for Medical Genomics. I have a bad news for the people who are sitting in, and that is the class that only registered students can go there, because that is a very small classroom over there. And that class will be taken at BRTC building. When we get there, we can give you the detailed locations of that. Okay, and we will also have a guest lecture at this point with Dr. Tatiana Freud regarding one specific biological applications. After that, we will have 3 lectures on the RNA sequencing data analysis, and including half a guest lecture from Milan Radovich, and to present his studies. And Dr. Bill Muro's lecture will be somewhere in October 28th, and he will tell us the power analysis of experimental design and many statistical issues that related to next generation sequencing data analysis. We'll have 3 lectures about chip sequencing, which also include the major application in chip sequencing epigenetics. And in the last lecture, November 18th, we'll talk about genome assembly, metagenomics, and 3rd generation sequencing and beyond. And then the December 22nd will be the student presentation, and after that will be the final exam. All right. Are we still good? Okay, let's get to the real lecture. So we got 20 minutes past already, talking about the logistics. And so today is the first lecture. So the topic here is a general overview of next generation sequencing technology. So I'm first introducing a little bit what is next generation sequencing technology, and I will go pretty detailed on some of the platforms. I want to give you a perspective why this technology can sequence that fast. What is the general biochemistry procedures on that? And I think it's an important topic for the students who are looking into jobs in this area, because once you go to the job interview, and a lot of data are sequencer specific, and you want to know how those sequencing has been done. And I will spend most of my effort here talking about the Illumina and Solid platforms, which are the 2 major ones that are in the research market. And I will spend half of the time to talk about the biological applications and the basic concepts and the challenges. So what is next generation sequencing technology? So why we do sequencing? Sequencing really means finding the order of nucleotides on a piece of DNA. So you can see this is the cell and nucleus, and we know the DNA is inside the nucleus. It's heavily packed very densely packed as chromosome, and it's very, very tiny. And however, if you really stretch it out, it's 2 meters long of DNA. It's very, it's amazing how the cells can make it. And we have 3 billions of DNA subunits. We also call it nucleotides, those ACGTs if you zoom in. And this will encode approximately 20 to 30,000 proteins and then eventually achieve their functionalities. So the sequencing really is trying to figure out what's the nucleotide, the order of nucleotides in a piece of DNA. The central dogma of molecular biology, and here is, I'll leave over. So we can see from DNA to RNA, from RNA to protein, and this protein will achieve their functionality. And this part is transcription, this part is translation. So we know that pretty well. And the nucleotide order determines amino acid sequence order, and by extension, also the protein functions. And alteration in a DNA sequence can lead to bad things. And the first one is if it's in the gene region, it can change the amino acid sequence and therefore change the function of the proteins, make it non-functional or less desired function. And then we are in trouble. Or if there's alternative is if the promoter sequence got changed, that can affect how this DNA transcribed into the RNA, affect gene regulation. So the end result of that will make more or less proteins. And if that's the problem, and we need to face. So the sequencing is the first step, trying to help us understand the genetic background of a certain disease. Hopefully, based on that, we can figure out the treatment. And also, I don't feel very, I'm not going to use this, and this kind of become annoying. All right, use this. So, so sequencing is very important. And now, in addition to biology and disease, and the sequencing technology has also been used in the forensic science and agriculture and a lot of other areas that are very important. Okay? And the history of the sequencing, and really it's an essential tool in the molecular biology toolkit, is ability to read the base sequence of DNA molecule. And there are several different technologies that has been invented, and the first rapid DNA sequencing in 1970s, and there's basically 2 categories. One is Gilbert and the Maximum using chemical sequencing, and the other one is very important, is the Sanger sequencing and using ddNTP. And I will have one slide to go over that a little bit. And from late 1990s to 2000, and there's a microarray technology has become very, very popular, very powerful. And this includes cDNA arrays and oligonucleotide arrays. And the major application of microarray technology is to do the genotyping on our DNA. So basically trying to figure out the genetic variations in one individual. Okay, go through a little bit of Sanger sequencing. I'm sure that if you're interested in this, you want to go to biochemistry textbook, and there are very nice introductions there. So the Sanger sequencing is using ddNTP, is D-deoxynucleotides, and it can be the ATP, CTP, GTP, or TTP. And these are the molecules that resemble the normal nucleotides, but doesn't have the normal hydroxy group. So you can see that these are the molecule, and this is the normal nucleotide. So, and this is the ddNTP. So the only difference between these two are this part, and in the normal nucleotide you got hydroxy group there, and in the ddNTP this hydroxy part is missing. It's only hydrogen there. So, and that serves as a terminator, so you cannot further put the nucleotides onto the DNA sequence. And by using this technology principle, that we can do sequencing, and that requires the template DNA. So you can see the template DNA. This is 5' end, 3' end. You do need a primer to get your sequencing started. And then you need a DNA polymerase to really put the nucleotides onto this, based on these templates. You need a pool of normal nucleotides. And also you need a proportion of labeled dd-NTP. So what happens is when you do the sequencing, and the polymerase will randomly pick a normal nucleotide or a dd-NTP and put on there. If it's normal one, it can go ahead to put more. If it's a dd-NTP, it's a labeled one, it will stop there. Let's see if we stopped here, and the next nucleotide, if happens to be a dd-ATP, we'll put here, not a normal nucleotide DATP, and the reaction will stop there. We know this molecule will become much shorter, so that on the gel, it will run much faster. So you can read this is A, right? And if it's a normal ATP here, and the next one you put a dd-CTP there, and it will terminate from there. So the length will be one nucleotide longer than the previous one. And when you run on the gel pictures, you can see that you put a C here. So the nice thing about this is after you do all these standard sequencing reactions, you can read your sequence from this end. And it basically represents the complementary strand of the DNA, the original template you want to sequence. Okay, and this is just a very brief overview, and if you're interested, if you're not familiar with this, and please check with your textbooks. Okay, and there are something about microarrays I want to talk about, and microarray has become really the equivalent words of high throughput technology probably about 10 years ago, and this is a very smart technology, and so let's go through that a little bit, and this is the surface of a microarray. So you can see there's a lot of small different compartments in the microarray. And this looks like a pretty dense. Yes, it is. Actually, for a normal Affymetrix array, there's over 1 million features on the array. And the size of that surface is the size of your thumb. So it's very, very tiny and very, very fine structures. And once you zoom in from here, and you will look like the things that will look like this. So for each different compartment here, you will put a lot of small oligos there, okay, a nucleotide. And this oligo sequence is designed in the way that where you want to target. So if you want to target a gene, you want to design these oligos reverse complementary to the gene you're trying to target. And then all the oligos in this small compartment will be the same. And for another compartment, you can target design oligos to target on different part of the genome. And when we do the reactions, and you can see that there's a lot of DNA, RNA molecules, and will be thrown into this environment. And all these molecules will be labeled. It can be fluorescent, could be biotin, depends on whatever technology you use. And once it's labeled, so you can see those green ones, molecules, they are perfectly reverse complementary to the probes of oligos on one of the compartment. It will hybridize to here, okay? And this purple one is not going to hybridize here because it's not reverse complementary to this part, okay? So by the end of the day, if there's a lot of signals in this area, I mean in the DNA you want to target on, and you will collect a lot of these labeled molecules, the signals. And it can be fluorescent signals, it can be biotin signals. Basically, you want to count how many small red hat are there. And the more you collect it, that means there's more signals detected in that part of the genome. As for this part, you can see there's nothing hybridized here. That means there's nothing expressed in those regions. So that is a basic principle of a microarray technology. It's kind of very smart. But how this microarray technology can help us to identify SNPs. And this is some technology called SNP arrays. Okay? And the SNPs are single nucleotide polymorphism, and those are most common genetic variations in the human genome. It occurred at about every 1,000 base pair in our 1 in every 1,000 base pair. Actually, it's more dense than that. Now we know it after 1,000 genome project, but that was the initial estimation of that. And when we go through that DNA sequencing topic, we'll go through all those different genetic variations. For the SNP arrays, and what it works is only for the known SNPs. So if you have a novel mutations or variants, you will not be able to detect that. So here is the principle of the SNP array targets. So you can see that this is the reference genome. Okay, this is the reference genome. And in this part, there's a SNP, a known SNP. It can be A or can be G, all right? So I got one people come in, and I want to know that is this person is A or G in this location, and that is the question that we want to answer. The way they design this array is, first they will design all these nucleotides are reverse complementary to the reference genome. You can see, right? Reverse complementary to reference genome. So T versus A and G is C out here. Okay. And at this particular location, they will put 4 different nucleotides here. So A, C, G, T. And 2 of them are mismatched because this is A. It's not going to hybridize reverse complementary to any of these. And G is a mismatch as well. T and C, because T can really reverse complementary to A, and the C is to G. So that is the perfect match probes. Okay, so once you put your DNA sample of one individual there, so if your pattern looks like this, and that will suggest that this is the same as the reference genome, and we know that it's a homozygous with the same as the reference, because this part, and which is the second probe, the signal is very strong, and the other 2 are not. Okay. And if this person is a heterozygous variation there, so you will see this type of patterns, and which the 2 mismatched probes, they don't have too much signal, but for those 2 perfect match, they both have reasonably strong signals. So that's an indication this is a heterozygous individual. And if the same thing, if it's a homozygous alternative allele, and you will see this type of pattern. So only the 3rd probe, you'll receive strong signals, and the other 3 not. Okay? Is that clear? Okay. So this is the SNP array technology, and again, it's only for known SNPs. Okay. And microarray technology has been very successful for about 10 years, and actually it's still very important technology so far. And there's many different popular applications, and which include the gene expression profiling and the microRNA or alternative splicing studies. So all these are on the RNA level. And on the DNA level, and you can using this technology to study DNA copy number variations or single nucleotide variations. And also there's another thing called the chip, so we can get into that a little bit in the later lectures. And this is a technology to study the protein-DNA interactions. And the disadvantage of microarray technology is also very dramatic, and actually next generation sequencing technology really help to fix most of this. The first disadvantage is the person must know the sequence to design the microarrays. So you have to know that where you target the sequence there so that you can design the probes to target that part of the genome. And we know that for a lot of expression arrays, for example, there's no way we can do that because we don't know genome really well. I mean, we assume that we know human genome pretty well, but actually there's a lot of non-coding regions. They are heavily transcribed, and we don't know there's important information there, so they are not put on the array. So the first disadvantage is you have to know it to design the array. And the second one is even sometimes you know the sequence, and you cannot really fit all the information into a small number of arrays. And the typical example will be the human tiling array. So the so-called tiling arrays, they try to design probes to target the entire human genome. And Affymetrix probably has the best technology there, and their platform is they have to use 7 arrays, 7 arrays to target the entire human genome. Actually, only half of them, the repetitive region, they are not going to target it. And the problem of that is 7 arrays and a lot of information there. But the problem is we don't use most of those information. So if you study protein-DNA interaction, we know that protein only binds on very small proportion of the DNA, and most of the information will be wasted. So that is a big issue for the array technology. And the third issue is the signal is not very accurate. It's a high noise level because of the cross-hybridization and the nonlinear information. So if you imagine that small microarray, so if this signal is very, very small, you probably won't accumulate enough signals for you to detect because there's not enough red hat there. But sometimes if the signal is very, very big in certain part of the genome, you'll also run into trouble because it can saturate all the probes in those regions. So it causes this kind of nonlinearity there. And those are big problems. Where for next generation sequencing technology, we just grab a molecule sequence, and those problems will be fixed. Okay? Any questions so far? Now here comes the next generation sequencing technology, and so one can sequence hundreds of millions of short sequences, and those short sequences range from 35 to 100 base pair, and in one single run. So we are not sequencing 1 or 2 molecules, it's hundreds of millions of those. And actually the latest technology, we got this high-seq technology from Illumina, and I think this is a 5000 XL platform from Solid, and they were both capable of sequencing 1 to 1.5 billion arrays in one sequencing run. That's a lot of information. And the major players that if you look into the literatures, you probably know that there's the Illumina technology, and that is the most famous one. And actually, it's initially called the Solaxa technology, and it was later purchased by Illumina. I think it was $600 million. All right, and the initial platform is GA, Genome Analyzer, and GA2, and now they comes up this high-seq machine, which is really, really powerful. And Life Technology, and it was ABI, Applied Bioscience, and they purchased the Solid technology, and also they recently purchased the Aaron Torrent technology. So both these two I'm going to talk about today. And actually the first, the real platform that is on the research market is probably 454 technology by Roche, and the headquarters, of course, is in Indianapolis, the U.S. headquarters. And the initial company was 454, and it was somewhere in Connecticut. I think it was purchased by Roche. This technology is very, very smart, and it's very important for a lot of biological applications. However, coming to today, if you're comparing that with the other two technology, the Illumina and the Solid, it's just too expensive. Okay, and when you talk about the pricing, and sometimes the sales rep gives the code, and they say, did you put one more zero in there? So it's that kind of a sense. So it's good for a lot of applications still, and it has its advantage. But for it's kind of for not competing very well with the other two platforms. And there's also other platforms, and like Helicos and Pacific Biosciences and Complete Genomics, and there's many such kind of platforms available right now. Okay, so there's so many different technologies. So what's the keywords of next generation sequencing technology? Some of you have already seen these slides in other classes. And the first keyword is sequencing. So basically, you want to sequence both the DNA or RNA. And RNA can be a modified protocol. And the second keyword is kind of important. It's a short arrays. Okay, you got this DNA sequence. It's not like you imagined we will sequence from the beginning to the end, just sequence it. Rather, it only sequence the very beginning of it. Okay, it depends on the platform. It can be 35 base pair. Actually, the earlier one is a 25 base pair. And 35 base pair up to 1,100 base pair with solid and the Illumina technologies, Selecta and Illumina these two technologies. But with 454, it's much longer. It goes to 400 BP. Okay, and each of this small thing you call it a read. And sometimes you can sequence both these two ends, and it's called either pair end or mate pairings, and that's very helpful for a lot of applications we will go through a little bit. The third keyword is ultra high throughput. It's really 1 to 1.5 billion reads per run. And for the 454 technology, you can see this is about 2 to 4 million reads. And it's 1,000 times smaller than the other two. However, their reads are much longer, so it certainly has its advantage. Okay. All right. Now I'm going to give you using an hour also to give you a brief overview of major platforms. I think this. If you are a biologist, you are not really interested in how these data are generated, just trust whatever has been sequenced. And this part is not that important, but I think for the students in the room, and you still want to know this. I'm going to just introduce a little bit detail on the Illumina and the solid technology, and I'll have brief things on the others. So the first one is Illumina Technology, and this is the most successful platform so far, and it probably holds about 60% to 70% of the research market right now. And so this is the slides for Illumina Genome Analyzer. It's not for the most advanced high-seq, but the principle, the biochemistry part, are similar. So if you look at the genome analyzer data, you will see that there's a concept called flow cell. So this is one flow cell, okay? And then you will have 8 lanes on this flow cell. So each part is a lane, okay? So in some sort, you can think this is just 8 different channels. Actually, you can treat them as independent experimental units, okay? 8 different lanes. So in a lot of situations that we put some human samples in 1, 2, 3, and mouse or other species sample in other lanes. So they are independent units. So it doesn't have to be for one biological applications. And one lane can give you about 10 to 30 million reads. Okay? So each read can be anywhere from 36 base pair to 100 base pair. And it has the capability to sequence a single end, which is only one end of the DNA fragments, or pair end. You can sequence both 2 ends of the fragments. And the pricing of this is for one lane. It's not the entire flow cell. It's about, initially it was about $2,000, and now the price dropped to about $800. So it's very affordable for a lot of applications. And also sometimes you may think, oh, I only study bacterial, and the genome is very, very small. I don't need 30 million arrays to study one sample. And actually there is an option that you can do multiplexing, meaning that you can put multiple samples into one lane and to share that kind of resource. And we'll get into that a little bit more. So now gets to the slides. Why do these technologies can sequence that fast. And the technology itself is really, really smart. So you can see that this Illumina is, the principle is a sequence by synthesis. I'm really not very comfortable with my neck breaks. So for both Illumina and solid technology, okay, there's two general steps regarding the sequencing. The first one is amplification. So if you got one sequence raise, it won't accumulate enough signal for you to sequence it. So you have to do the amplification. And the second step is to do the sequencing itself, to read the order of nucleotides. So the first two slides of this, it tells you the amplification procedure. So you can see that this is the DNA fragments you really want to sequence, okay? And these are some sort of adapters. And those are from your sample, and these are not from your sample. This is from library preparation kits, okay? The first step you do is you use ligate adapters to the end of the DNA fragments you want to sequence. So you can see that these are the part that you are interested in what the sequence is, and these are the adapters you already know what the sequence is. And the second step here is you want to attach this DNA into your surface. So you can see that there is a adapter and the DNA fragments and the adapter. So this is the whole thing. It will stay on this surface, okay? And there's another one stay here, another one stay here. But a lot of others are just those adapters. They call it the adapter lawn, okay, like a grassland. And the next step is that they will do something very smart called the bridge amplification. So these molecules will be banded here, and to touch to whatever the adapters, it will match in the surrounding area. So you can see there's a lot of other adapters around here. And then you can use the amplification procedures to do the amplification for that part. So this is called the bridge amplification. And after you do that, and each bridge, you can see this is attached. So this part of adapter is attached to the surface, and there's a free terminals as well. So these are not. And then you can detach it, and so it extends again. And then you can follow another cycle of bridge amplification, and that will eventually give you a cluster of those sequences that you initially put on the surface. Okay, so this is something they call the bridge amplification. All right, are we still okay? Okay. And I know these colors are not the best for you to read from pretty far behind, but I didn't make those colors, okay? I just got those from the Illumina website, so let's blame them, okay? So this is the first part of the Illumina technology. So basically, you want to amplify your DNA molecules and further do the sequencing. And now comes to how we do the sequence. It's really sequencing by synthesis, okay? And what you do here is, remember, after bridge amplification, we will have so many clusters of DNA molecules, okay, and standing there. And you will flush in a lot of labeled nucleotides, and which are all labeled in different colors, A, C, G, T, 4 different colors. And then once it's attached to here, so you can see this one, the first one is green, yellow, okay? Pretend to be colorblind. This is yellow, and this is green, and this is blue, this is red. And then you will take a really good picture on the slides at this moment. So at this location here, it's yellow, so you will see, oh, there's a G there. Okay, the first base is G for this particular cluster, and the CAT. It depends on the colors, okay? And next step you are going to do is you want to determine the second base, and there's some chemicals you can wash the first base away, and then further do the synthesis. And then the second color, you can see this part is become a blue. it was yellow for the first base, and second is blue, and this is still red and with different colors, okay? And then you know that once you got it, the first picture you take, this location is yellow. The second picture, which represents the second base at the same location, it becomes blue and the green and the yellow. So you read the colors, you know the sequence at this cluster will be GCTGA, and just based on reading these colors. Okay? Does that make sense? Okay, so there's a lot of details, of course, you can go through, and which I don't understand either. But you can really look at that, and the first part is amplification. For Illumina platform, this is the bridge amplification. And the second part is how you detect the signals, which is using this colored nucleotides, and then you want to take pictures while they are being synthesized. And the current platform of Illumina is HiSeq, and it's 1 billion, you can generate 1 billion clusters, okay? On that small surface, you can generate 1 billion clusters, and then, therefore, each cluster will represent one DNA molecule you want to sequence. And how much information is 1 billion clusters? It can cover the human genome twice with a 30x coverage. So this is what I meant. One technician in 1 week, one machine, and you can sequence the human genome twice with very reasonable coverage, with the cost about $10,000 per sample. Okay, and there's a multiple. It depends on the needs for the application. Sometimes you only need a single end, which is 35 base pair. It can be longer to 75 or 100 base pair. Or you can sequence that pair ends or mate pair, so both two ends of the DNA molecule, and each one can be 50 base pair to 100 base pair. Of course, the longer you want to sequence and the more time you will need. You may say, okay, I'm fine, I can wait. And for another 4 days, but this is really equivalent to the cost, right? The real cost for this sequencing is the reagent cost. So if you use 8 days for the reagent, and you really double the cost at that point, at least the sequencing part of it. And there will be a lot of data can be generated. Okay, so next, I want to turn around to a little bit of a solid procedure. Solid is another very important platform that you will have to be familiar with. There's a couple of reasons, and when we talk about the market shares, you will see that Solid is such a major player. And another thing is the data is not normal. It's kind of confusing to begin with. So let's go through that. And comparing to the Illumina procedure, the solid and the amplification part and the signal detection part are both different. Okay? For the amplification part, instead of doing bridge amplification, you do something called emotion PCR. We'll get into that a little bit. For the signal detection part, it's no longer done by the polymerase-driven incorporation of labeled nucleotides. Rather, it will use a mixture of labeled oligonucleotides and queries the input strand with ligase. So I will tell you what I mean by that. In the short words, this is a sequence by ligation. Okay? And another major difference between Solid and the Illumina platform is Solid gives you the color space, not the base space. Okay, so here's what I mean. Remember, in the Illumina technology, we have A, C, G, T. We have 4 different colors. Okay, it's very straightforward. For Solid, it doesn't look like that. So what happens is for AA dinucleotides, CC, GG, TT dinucleotides, they will give you blue. Okay, and for the AT, they will give red. CG, they will give red. So you can see that every dinucleotide, they will give you a color Of course, we have 16 dinucleotides, right? A, A, C, A, G, A, T, or whatever it is. But there's only 4 colors. So this is how the experimental design is done. So and it doesn't really give you a straight answer of what the sequence is by the end of the day. Rather, it gives you a lot of 0 and 1s, which make you very much confused. Okay, and when we talk about, I will have in the later slides, and this will give you a lot of good things as well, and including the quality controls, which is very important. And here's the good thing: is each base actually is sequenced not once, but twice. Okay, when we get into that, you will see what I mean by that. And so this is really one thing that makes the solid sequencing very accurate. Okay, and because every single nucleotide has been sequenced twice. So if you imagine Illumina technology, the sequencing rate, error rate is 1%, and for solid, it can go to 1% of 1%. Right? Because every single nucleotide has been sequenced twice, and that is really good for the quality control part. And it does have the built-in error tracking capacity that distinguish between measurement error and true polymorphism. Okay, we'll get into that, and the detection more complicated variations. Okay, here's how it works. Okay, so remember I talked about there's 2 parts on the high throughput sequencers. One is the amplification part. The second is the signal detection part. Okay, the amplification part is something called emotion PCR. So what happens is these are the components that are required for emotion PCR. You will have these beads, small beads, and with a lot of adapters attached to it. And you also have this DNA molecule that you want to sequence, which is the blue part here. And you have a lot of primers which help you to go through those sequences. Okay? And so this is the basic components. And this is the unique part of it. It makes the PCR allocase phase into a water in oil emulsion. Okay? So each part you can imagine this is a bubble, a liquid bubble, which trapped your bead and the nucleotide you want to sequence. All right? And what you want to have is for each bubble, you trapped 1 bead and 1 sequence in there. And sometimes you trapped 2 beads and 1 sequence. It's fine too. It's okay. But you might have trouble when you trapped 1 bead and 2 sequences there. And those are something that will eventually cause trouble. But the point here is, and for each one, you can imagine this is 1 small experimental unit, and it's in the bubble of a liquid. Okay, within each bead, within each bubble, and you still got this bead, okay? And then what you have is this will hybridize this linker, but the end of the DNA molecule will hybridize to this bead, okay? And it will stick out. So you can see now we still use polymerase to do the amplification and eventually get 2 nucleotides here, another one here, okay? And then you will come back to this single base. So by denature this double helix here. And this is what happens in one of these bead, in one of these beads. But after you do this PCR in many, many cycles, and you will get something like this. So you can see that each bead have a lot of, so each of these adapters originally, and has the same identical molecule growed around it. So you can think this has become a ball there. And now at this point, those small bubbles are not very much useful anymore, and you can break it, and then it becomes like this type. So you can see there's some bead that has the same molecule that just grow around it, and this is a different molecule. But some of them, which one? There's one of them are not very good. So this one has 2 molecules grow around that, and there's also a lot of bead that doesn't have anything. Okay, And then you break out these things, and then, so you can see there's basically 3 types of beads after this emotion PCR reaction. The first one is the usable bead. Okay, this is the bead contains about 30,000 amplified products from original single-strand molecule. Okay, you remember that we initially have only one DNA molecule attached to it, and after emotion PCR, we got 30,000 of those. That provide enough molecules for you to do the future reactions. And there's two type of beads that will be removed here. So one type is there's nothing attached to it initially, so you don't really have anything growed out. And there's some others that cause the sequencer to be confused because there's 2 type of templates attached to it. You want to get rid of those. Next step is you get this perfect bead, and then you want to also attach the addition of a chemical group that will eventually allow this bead to grow on the surface. Okay, and this is what happens. So you got so many beads, they are already perfect. They have all the DNA molecules grown after the emotion PCR, and you dump it on this flat surface. And this surface, these red dots we just put there, can help these beads to attach to those surfaces and put beads on the slide. So you can see this is what they look like. Okay? And actually, the throughput of this sequencer, it really depends on the size of the bead. The smaller the bead is, the more dense they can really pack it on the surface. And also the density of the beads eventually on this surface. And that's something they want to keep improving and therefore get more high throughput coming along. Okay, so this is only for, it's not for the sequencing part yet. It's only for the amplification part, which is the emotion PCR. Okay, is that clear? Now we try to see how do we do the signal detection here. Okay, so pay attention to this part because this will tell you why we got a color space in the solid protocol. Okay, so before we get into that, this is the tool that we use to do the sequencing. So you can see it's very, very complicated comparing to Illumina technology, which is a nucleotides and you just put in there. So what happens is that we call it a probe. So you can see that each probe have this 8 nucleotides, okay? And we'll go through these 8 nucleotides, and also attach a specific color by the end of the nucleotide. And these 8 nucleotides are 2 nucleotides that you want to sequence. You want to target, you know what they are. And there's 3 nucleotides, they are actually degenerate bases. So basically, it can be anything, all right? Oh. Don't bother with that. Okay, and there's also 3 nucleotides. They're universal base, so they can hybridize to anything. Okay, so in total, only the first 5 nucleotides are important because these are universal ones. And these 5 nucleotides, and there's a totally 1,024 probes because we have 5 bases. Each one has 4 different choices. And actually, we have 4 dinucleotides, and because there's 4 different colors. And so if this is the first 2 nucleotides is TC, so if we look at this TC, and we will put the yellow dye here. Okay, if it's AA, we'll put blue dye here. So you can design this type of thing. Just see, I'll just let them go. So. Okay, so each nucleotide is each dinucleotide is encoded by a color. And this is how sequence goes. Okay, we first will get 1 bead. So you can see this is the bead, and this is the P1 adapter, and this is the template you really want to sequence. Okay, and the first thing you want to get here is you will do the ligation reaction process. So you can see that this TA, it will reverse complementary to this 5 nucleotides. It will reverse complementary to here, and it will attach to here, and we ligate them together. Okay, and because this file base is really hybridized to whatever the sequence is, and you can read the colors, and then to know that what is your first dye base the color is. Okay, and the next step is you want to take a picture. Okay, take a picture at this point because of this green signal. This one is attached to here, and the green signal, you will know that first dye base is green. Okay, and you probably cannot read it from back, and actually it's, the sequence is TA. And then next step is you want to cleavage this. So you can see that this fluorescent signal together with these 3 universal rays and will be break out from here, and this color will go. And next step is you do the second cycle of ligation, and you start from here. And now you can kind of have this gray signal here, and you will know that what is your second dye base is. So this is the number 1, number 2, and number 6 and 7 nucleotides, and this dye base will be sequenced as well. And then you take a picture, and you know what the second dye base is. Okay, and now you reset one, and you do this many times, and then get interacting covered. Okay, so keep in mind that there's only the position 1, 2 has been read out, and this 2 position has been read out, and this 2 position. So there's a lot of empty base in the middle, so you haven't sequenced it yet. And then what happens is you will get rid of this primer as well, and then get in another primer, which is a primer N-1. So you can see this is a primer N, and N-1 actually is just 1 base pair into the primer, the adapter, and then you redo this process again. So we know this part is T, so this is AT, and you will see this is a red one. And now you take another picture, and then you know what this 2 dye base is. Okay, and you probably are still confused, and I hope this figure will help you to understand this. So for the first universal sequence primer, the first one, and then we will first do the ligation of this part, so you will know these 2 dye base, what the color is. Okay, and you will know what the color of these 2 is, what different color of these regions are. And for the second reaction sequence site, so you go back, you will actually look at what the dye base of these 2 colors are, and these 2, these 2, these 2. And then you will change your primer again, and then you will target this part, and this part, this part. So using 5 different universal sequence primers, by adjusting their starting locations, you will know that every single nucleotide has been interrogated twice. So other than this one. And so you can see this one has been sequenced twice here, this one be sequenced twice here. So every single nucleotide has been sequenced twice. Okay? And then you will, by the end of the day, you will know for what is 0, 1, 2, 3 are. So by their dye base composition. Still confused? Think about it. It takes a while to think it through, okay? And so if you think the biochemistry part can be confusing, and the data analysis part can be even more confusing, right? And so you have to get familiar with this color space issue. So you can see that we know the design of these colors are AA is blue, and these are different colors with different dye bases. Okay, so if this is your DNA sequence, and we AC, okay, if we see AC, this is green, and we'll report it as 1 here. Next one, next one is CG. So CG, and it's red, it's 3 here. So you can really calculate this color space and from the original nucleotide sequences. Okay, based on just if you know what the first one is, and you should be able to derive whatever the color space should be. The problem is we don't know the sequence of nucleotide. That's something we want to sequence, all right? We will be able to read the color sequence. So we'll be able to know what this sequence is, okay? We will not know what this is. But if I know what the first letter is, I can really derive what the other letters are based on these color space sequences. Does that make sense? All right. So there's some properties of this dibase coding, and the first one is it's kind of interesting. 2 dibase that agree in just one base have different colors. So A, C, AG, AT, AA, they are all different colors, which means the first row, for each row and column, they are all different bases. The second property here is 2 dibase that do not agree in either base have same color. So you can see this AC and GT are always the same color. And CG and AT are always same color. You can go back to this to figure that out. And the third one is a dibase and reverse have the same color, which is very nice. AC and CA have the same color. So if you can look at that. And repeat base dibases have the same color. So all the diagonals here are the same color. Okay? So these are some of the properties of this color space. You are still confused. I know that. And so we have to give you some incentive why this dibase is good for us. Okay? And let's take a look at this particular example. So you can see that this is the sequence, and this is another sequence. Let's see, imagine this is the reference sequence, and this is the sequence you get from what sample you got, right? And let's pretend we already know what the sequence is, okay? And we also know at this particular location, there's a SNP here. It's a single nucleotide difference between the reference and the alternative. Okay, so you can go through these colors, and then you will find a very interesting feature for single nucleotide variation is every place are the same color with a single nucleotide variation. However, with a single nucleotide difference, they have to have 2 dibase, 2 color are different with each other, and that becomes a single nucleotide variation. Okay? This is something that built into this color space. If you look at this one, okay, every single base are the same color, with only one base are different. One color is different. And this doesn't align really well, because once you derive, based on the color, derive the sequences, from this point on, every single nucleotide will be different from what the original one is. So what that means is if there's only one color difference between the reference and your sequences, this must be a sequencing error. Okay? It's impossible to be a single nucleotide polymorphism. And having this extended is if there's one variation here and there's a valid changes of their colors, and you can see there's both 2 colors are different. If you have 2 color changes, and this it's a valid single nucleotide variation. If it's one color change, regardless how you play with the colors, this is an invalid change. This is sequencing error. And some of the 2 color changes are invalid as well. So the point is, if you build your algorithm smart enough, and this is built-in check of the quality of your sequencing. you know it. It is based on the number of changes you observe here. You already know it's a sequencing error, a single nucleotide polymorphism. Okay? So I encourage you to go back and to sit down, really think through this, and if you cannot, that's what the office hours are designed for, right? And but this is a very interesting, very good feature. So it's very unlikely that two consecutive bases they have the sequencing error. It's very unlikely. If one nucleotide the sequence is 1%, and the two both have the problem, and the problem is the probability is 1 of the 1%. So this is really help you to increase your sequencing quality dramatically. So the summary for the solid platform is there's an advantage, which is a two-base encoding provides higher system accuracy. Each base is sequenced twice. The probability that both of them are wrong are different. And it also has the built-in error checking capacity. So if you see two consecutive color different from reference genome, that's a real one. If only one changed, that's a sequencing error. So you already know that. And some of these color space rules, I'll just talk about alignment. Some of these rules can also be applied to detect error during the de novo assembly, which we are not going through too much. We will not go through too much. All right, I think we can have a break here, and we'll come back in about 10 minutes. All right.